Pouya Kheradpour on systematic prediction and validation of enhanced regulatory motifs in ENCODE cell lines.
Okay, thank you. Thank you for this opportunity today to talk about our systematic prediction of and validation of regulatory motifs, so dissection of regulatory motifs and enhancers. First, I'm going to talk about how do we predict activator and repressor motifs in cell lines. Then I'm going to talk about how do we disrupting these motifs to see which ones are functional and which ones aren't and how they work. And then specific experimental results on activators, repressors, and their context. And finally, what happens when you swap motifs between contexts. Okay, so we have a pipeline for discovering motif instances using conservation, and we're going to use this throughout this work, using conservation throughout the eutherian mammals. And we also use chromatin mark-based cell line specific enhancers, which Jason will talk about more tomorrow. But basically here, what we have is a set of enhancers that are specific to H1ES cells. And if we look at enrichment of motifs, so here the red indicates that OCT4, the motif for OCT4 is enriched in these H1ES enhancers, and likewise OCT4, the transcription factor is also expressed in these H1ES cells. Conversely, what we see for GFI1 is that it is expressed in K562 in GM cells, but it is the motif is depleted. So what we would predict here is that OCT4 is an activator of H1ES in H1ES, whereas GFI1 is a repressor in K562 in GM. And of course, we have more than just 3 cell types. We have many cell types here. Looking at 9, we see that we predict a number of activators and repressors for each of these cell types. And I want to draw your attention specifically to these 2 clusters, these HepG2 enhancers. And we see that we find activators HNF1 and HNF4. So we've moved this beyond this, and we are now applying it to DNase data from John Stam's lab. And we find that basically these same types of methods work there to find activators and repressors across hundreds of cell types. Okay, so now we wanted to test these predictions and their motif instances specifically. So here we ended up predicting 5 activators for these 2 cell lines, HepG2 and K562. So these 3 for HepG2 and these 2 for K562. And this is based again off the expression of the factor and enrichment of the motif. And we also predicted a repressor for each cell line. And the way we tested these is by taking instances of these motifs inside 145 base pair context within these cell line specific enhancers, and then performing various manipulations of them, so either scrambling the motif, removing the motif, or doing single nucleotide changes, either to decrease the motif match score, to keep it the same, or to increase the motif match score, and then also 2 random changes. We took these and synthesized them on an adjuvant array, and along with a tag, and then inserted an SV40 promoter and inert ORF in between, and transfected them in K562 and HepG2 cells, and then counted the tags to produce basically a digital expression value for each sequence in each cell line. Okay, so the results that we saw, so looking at a single example, we chose an HNF4 motif instance that was conserved within a HepG2 enhancer. So this was criteria for choosing this sequence to test. And we found that it was also coincident on a H3K27 acetyl dip, which is indicative of perhaps a nucleosome exclusion. And when we tested this sequence, what we found is robust expression in HepG2, but no expression in K562. So this line here is kind of like the average. And now when we do the manipulations, so if we scramble, remove, or do the best possible decrease of the motif, so here taking the G and changing it to a C, what we see is that the expression is basically abolished. Conversely, when we do changes that we don't expect to influence the motif, so here, for example, taking this G, which is not particularly important, and making it a T or slightly improving it to a G, so it's originally an A, we see the expression either stays the same or maybe slightly increases. And the 2 random changes we do in this case have the effect that we expect. So in particular, taking this T, which is not very important, making it to a C has a small effect, maybe it increases a little bit, whereas taking this important T and mutating it to an A reduces the expression dramatically. That's just one example. Of course, we tested hundreds of sequences, and here for the conserved HNF4 motif instances, we see that all the disruptive changes are basically below this line, or almost all of them, whereas the non-disruptive changes are on the line, where here we're showing the expression of the original sequence versus the expression of the modified sequence. And so this is one factor of the 5 factors that we tested. So here you see, so for HNF1, HNF4, and FOXA, which were HepG2, picked in HepG2 enhancers and tested in HepG2, we see the expression of the highly conserved motif instances is higher than if we don't use conservation in choosing our motif instances. And then both of them are basically much higher than when we do the scrambles, which effectively abolishes expression. And we see the same thing for NK562 with GATA and NRF2. And these p-values are all very significant, as you can see there. We also tested these other manipulations, but we only did a handful per factor. So if we look at the combined here, we see that the trend is what we expect. Again, with the disruptive mutations abolishing activity, the neutral change leading to basically no change in expression. The p-value is not significant here, actually. And then the increase, or the improvement of the motif match actually leads to a significant increase in expression. Okay, so one surprise that we saw is that we saw that NRF2 was also functional HepG2 cells, which we did not predict. And so this was a little bit of a surprise. And it suggests that maybe this sort of approach can be used to find functional enhancers in these different cell lines, or functional factors in these cell lines that might not be showing particularly high expression or even enrichment of motifs. We also looked at the 2 repressors. So, for example, for GFI1, we tested GFI1 motif instances and HepG2 enhancers with the hypothesis that while GFI1 is expressed in K562, it might be turning this region off. And by testing this, we'll see the expression actually go up. And we do in this example. So you see this expression is lower than baseline in K562, and then it increases upon removal of the GFI binding site, suggesting that, again, maybe this enhancer would be on in K562 were it not for this binding site that was turning it off. And when we look at all of them, we do see a significant increase for GFI1 here. We don't see it for CFP161, and we don't expect the same level of increase or difference as we do for the activators, because we don't expect necessarily that all these enhancers that are on in one cell type are primed for the other one, except for this repressor site. But it adds an interesting dimension to the analysis. And finally, we were interested in finding out, well, what made these activator motifs work in the first place? What was it beyond just the motif? What was in the context of the sequence? We found a number of features that were correlated with activity. In particular, this nucleosome exclusion that's captured by this DIP score and the DNA hypersensitivity overlaps, we found were very significantly associated with whether or not the enhancer worked, even though the plasmids that are being tested are not necessarily picking up these chromatin marks. And finally, we found that motif conservation, as I showed before, the number of factors and the motif match score were also important. But one thing to highlight here is that the motif match score wasn't everything. And so it's not that the stronger the motif match score, the higher the level of expression, or the more likely that it's expressed. And so it's clear that the context is very important. So we set out to test this specifically, so by swapping the motif between enhancer contexts. Okay, so the experimental design we used here is that we took the top 10 motif matches, or top 10 enhancer sequences for each of these 4 motifs, so HNF1, HNF4, GATA, and NRF2, in the context where we expected them to be functional from the previous design, and we basically swapped the motif instance between them. So, for example, we would take the original sequence, we would reverse complement this motif, and we would swap in a motif from another one. So here, taking the HNF4 motif instance from one of these HNF4s and putting it in the HNF1 context with 0 padding, or maybe adding some padding around the motif. And we also use a scramble motif, which I use for normalization here. So what do we see? Well, one thing that we could look at here is what happens if we just swap the motif instance amongst the the same factor. So taking, for example, all the 10 HNF4 motifs and swapping them between them. And what we found is that the wild-type sequence was significantly higher than any of the other ones, which suggests that the motif is somehow tied to the specific sequence that it's being found in. Perhaps there's other binding sites or something that overlap with the motif that are important in this context. And moreover, although you can't clearly see it here, the sense strand was more important than had higher expression than the anti-sense strand, which again suggests that maybe while the motif itself and maybe even the enhancer doesn't have a sense with respect to the gene that is regulating, within the enhancer itself, the sense may be important. Okay, but what may be more interesting is what happens when we swap from one context to another. So here, looking at HNF1, which we expect, which we find is active and expressed in HepG2, taking HNF1 enhancers with their motifs swapped around between them, we find the expression is quite low in K562, which is not a surprise. But as we incorporate the GATA motif, which is functional in K562, we see the expression go up, and this continues as we increase the expression level. Here are the dashed lines showing the reverse complement. And finally, the original motif in its original context is quite high. And this is a trend that we see for all these pairs that we test, where we go from one cell type to another, and you see they all kind of increase. And this is in contrast to what happens if we switch within the cell type. So switching between NRF2 and GATA, which we expect are both functional in K562, we see the expression is more or less flat. And this suggests that the context might be tied not necessarily to a specific factor, but more to the cell line. So you need a context that's receptive to the cell line, and then the motif match to also be receptive. Okay. So in conclusion, we tested thousands of sequences and now have hundreds of experimentally validated enhancers with the necessary motif instance identified. And this could be a resource for data mining enhancer sequence elements. There's also certain general principles of activator and repressor motifs that we have either confirmed or developed here. So the motif is very important, and in fact, in the cases that we saw, generally mutating the motif disrupted, or disrupting the motif abolished all enhancer activity. Moreover, several contextual clues that we found were important, including conservation, nucleosome exclusion, the binding the other TFs, and the motif strength, and even the cell line of the context. So again, not that you have a generic enhancer context where the correct motif will cause expression as you expect, but rather the cell line is tied to the context specifically, and then also the importance of repressors from what we saw with GFI1. And moreover, we have a general methodology for testing enhancers. So all this work was supervised by Manolis Kellis, who's my advisor. Tarjei developed MPRA along with his group and others at the Broad, so Alexandre, Peter, Lee, Charlene, and Jessica. Jason developed the chromatin states, which I used, and he'll be presenting tomorrow about them. The chromatin data itself came from Brad Bernstein's group, and Alex and I developed the motif instances that I spoke about. Thank you.
So good work. So I have two questions about your work. One is that in your motif analysis, have you found any motifs that are specifically attached to the enhancers, which are distinguished from those that are identified in the promoters? And the second one is.
Sorry, what was that? So you're asking.
I mean, do you find anything any motifs that's specifically targeting enhancer regions rather than promoter regions? Because that might suggest that there is something unique about these factors. I guess the second question is that when you do this validation experiments, you shuffle these sequences. I was very curious. Do you see any position effects, for example, like if you move these motifs like closer or farther away from particular area? I mean, do you see any differences at all?
Okay, yeah. So I'll start with the second question because I can just tell you we haven't done that experiment yet. I think that's an interesting experiment. So, but combinatorially it becomes quite complicated. So we've already the second experiment I talked about tested 12,000 sequences. So doing a position effect, while very interesting, I think would be worthwhile to do, would be complicated. As far as the The first question, can you remind me what it was again? I'm sorry.
So the question
Oh,
is
promoter,
that,
yeah, so we haven't looked at this specifically in this context. We do find that, in general, the same kinds of patterns, like we would see, HNF1 motif enriched maybe in genes that are expressed in liver, but promoters tend to be, they're not as dynamic as enhancers, and so I think it's a little bit more muddled what you end up with. So there's a lot more promoters that are kind of active in many cell types, whereas enhancers tend to be very cell type specific.
I'm not asking about the dynamics. I'm just asking, in the distribution of the motifs, are they primarily biased toward the enhancer regions versus the promoter regions?
No. So we don't.
This is different kind of classes of.
No. We definitely see a lot of enrichment in promoters as well. Although in this study specifically, we only tested enhancer. We specifically excluded things that were close to promoters.
So very nice talk. So you mentioned NRF2. That pathway has been shown to be altered in 40% of lung cancer by the TCGA work. So have you mined that data to see whether you saw any mutation in the enhancers of the NRF2 factor in the primary patient data?
No, we haven't. That's a good idea. We should follow that up. Thank you.
Okay, the next talk.